Okay, to start off this lab, we use sharp scissors and the forceps tweezers to create a slit in the adult sea squirt in order to be able to get it out of its bubble to be able to identify the chordate characteristics. And then as you can see in the video, we switched from sharp scissors to tweezers to pull it out of the slit that we had made. For reference, here is a photo of an adult tunicate or sea squirt. Its body plan, and here you can see the in siphon and the out siphon, and the stomach and the intestines, where the water goes in through that in siphon, through the stomach and intestines, and out through that out siphon or excellent siphon. And we are going to be trying to identify these things from this photo on the organism that we saw in lab. And here is a photo that we captured in lab. Once we removed the tunicate from its membrane, we were able to find the intestines pretty quickly because they're large and take up a big portion of it. That would be indicated by that bottom gray arrow on the image. And then based on the location, we can conclude that the siphon that's indicated by the blue arrow would be the out siphon because it's so close to the intestines, which would lead us to believe that the siphon indicated by the red arrow would be the in siphon, where the tunicate takes water and nutrients in. What we have here is a general body plan of the larval tunicate or a tadpole. And here you can see the 5 chordate characteristics, being the notochord, nerve cord, the pharyngeal gill slits. It's kind of cut off, but there is a line at the top pointing to that tail, the postanal tail. And then there is not an arrow pointing to where the endostyle would be on this organism, but it does contain one. And this is the larval tunicate. As we saw before, you don't necessarily see all of these characteristics in the adult stage, but it still would be classified as a chordate because of the fact that it had the characteristics in its larval stage at some point in its life. Here is what we saw in lab, the actual slide of the larval tunicate, and we just highlighted what the generic photo that I just showed before showed. The green arrow points to that postanal tail, and then we didn't really point out the notochord and the nerve cord just because they are a little bit harder to see from this angle. And then the red arrow on top to the right is pointing to the gill slits. And then something interesting, the 2 bottom arrows are pointing to the siphons, the in siphon and the out siphon, with the in one being on the right and then the out one being on the left. And that just is a little bit interesting because you can still see the siphons through to the adult stage. But there aren't very many chordate characteristics that the adult tunicate continues to have besides an endostyle, really. So it's interesting also that the larval stage has all of the characteristics and then eventually loses it throughout the lifetime. What you see here is the Amphioxus slide or the sea lancelet that we looked at in lab. And there are different ways that you could look at this slide, but the way that we set it up, we focused on this forward or the front part rather than the back end, just so that we could more closely see more characteristics, because on the back end you just really see the tail, the postanal tail. But here in this image, the way that we set the slide up, you can see that notochord and the nerve cord, the thicker part of that back there. And then all of the little lines, the pink lines that you can kind of see through, those are the pharyngeal slits. And then this organism does also have an endostyle. So it's very similar. It has all of the same characteristics that the larval tunicate or the tadpole had. And so this one also would be considered a chordate. It's just another way, another representation of those characteristics. And now we have a slide of the Ammocetes. And we are going to compare this one to the larval tunicate and the Amphioxus that we just looked at. This one has all the chordate characteristics, and we also set the slide up the same way. We noticed that this end seemed to be the fronter cerebral end, where you could see more of the characteristics. If you were to focus on the other end, you would just see that post-anal tail again. And then where this organism differs from the Amphioxus and the tadpole is that it is more specialized, has more sensory organs, more developed sensory organs in the cerebral area, but obviously, as you can see, it hasn't really developed a brain yet. You can't actually see a cranium or a brain. But you can see in the nerve cord and nodal cord region that it connects to the head, which we can use to make the deduction that this organism actually is a vertebrate. So it has all the same chordate characteristics as the Amphioxus and the tadpole, but you can actually tell that it will develop a spinal cord, and that would make it a vertebrate because one of the big characteristics of vertebrates are those vertebral columns and cranium. Again, we can't really see the cranium at all, but it definitely looks more specialized in that region. You can kind of get the hint that this organism is a little bit more developed than the two that we just looked at before. And then just as a refresher, this is what that Amphioxus looked like one more time, the sea lancelet, and you can tell that the nodal cord and the nerve cord just end in what we assume to be the anterior region, but there is no sign of specialization or anything of that nature at all. Moving on, here we have a cross-section of a lamprey. We cut ours in half in lab, so it does kind of look like a mirror image. And the lamprey actually is in the adult stage, and the Ammocetes was the larval stage of this organism. So as you can imagine, it does have all 5 of those chordate characteristics, as well as the fact that it is a vertebrate. We just have a few of the chordate characteristics highlighted here. As you can see on the right with the pink arrow is pointing to those pharyngeal gill slits. They do appear to be small holes in the organism. They look a little bit different than the slides that we looked at before. And then that blue arrow on the left is pointing to the nerve cord, and then the yellow arrow is pointing to, of course, the nodal cord, which sits on top and protects the nerve cord. And then not shown in this image is the postanal tail, just because the lamprey was a bit large and was hard to get it all in one photo. But we thought it would be best to get the majority of the characteristics in this top half. And something fun and interesting is that our lamprey was a female. That's what all of the pink looking stuff is. You can kind of see it behind the blue arrow. On both sides, those are eggs. So you can tell that our lamprey was a female because of the fact that she had some eggs in her still. And then this organism does also have an endostyle. And you can tell how advanced this organism gets to be in its adult stages. Very more complex than the adult tunicate that we looked at before with the siphons and the intestines. Yes, and then you can kind of see the brain closer towards the front of the head. Unfortunately, there's not an arrow pointing to it, but yeah. So that's how we can tell that this one was a vertebrate, because of the fact that it had the brain and the cranium and developed the vertebral column as well. All right, now to wrap up our lab video, we've included a cute, funny photo of a vertebrate, kind of relevant to what we were talking about today. It definitely has a spinal column, a vertebral column. um, so just to wrap up, we're going to talk about why we study protochordates or invertebrates today. And that one isn't too hard. Protochordates are that halfway point between chordates and invertebrates. And they can provide, or studying them can provide some really good insight on evolutionary history, which organisms are most closely related to which, basically how we got from one point to the other and how we ended up being the vertebrates that we are today. And yeah, researchers can study them and see which small changes, adaptations occurred, and how the development changed and advanced throughout the phylogenetic history. So they are pretty important in understanding how chordates turned into vertebrates, kind of, one hypothesis. And yeah.